Welcome to the Human 5.0 Project, the podcast where we cut straight to the breakthroughs that matter, the show where science meets self-optimization. I'm your host, Ultimate Biohacker 10X, and here we break down the real research, real data, and real dosing behind modern biohacking, from compounds and peptides to techniques, devices, lifestyle strategies, and performance protocols. Most people jump in and jump out before they get the gold, so we're keeping it tight and delivering the essential insights you need without the fluff. If you've taken the time to hit play, we'll make sure you walk away with valuable knowledge that can transform your approach to longevity, health optimization, and beyond. I was going to wrap it up early today, but I've been thinking a lot about exosomes and stem cells and conversations that I had today, and the more I thought about it, the more I felt like I needed to add on to the exosome talk that I did earlier. And so we're going to blast another one out right now, which is highly valuable, I think, as an adjunct to the previous discussion. And it will probably bring up some other interesting comments as we move forward. So I just wanted to give this quick intro and let you know why we were doing another podcast following up the one that we just released. All right, so let's talk about one of the slickest claims floating around regenerative clinics right now, that stem cell-derived exosomes can be sterilized, freeze-dried into a powder, stored at room temperature, reconstituted with saline, and injected like nothing happened. Sounds convenient? Sounds modern. Sounds too good to be true. Today we're going to do a 2026 reality check, what the biology says, what the human data actually shows, and why convenience claims can quietly put patients at risk. Loyalists, biohackers, and longevity rebels, this is Ultimate Biohacker 10X firing up the mic one last time today, and we are here to stretch your timeline, protect your cells, and keep you from mistaking marketing for medicine. Welcome back to the Human 5.0 Project. This episode is for educational purposes only and is not medical advice. As of 2026, there are no FDA-approved exosome products for disease treatment. Many products marketed in clinics or online are unapproved biologics. Safety, dosing, purity, and long-term effects are not fully established. Always consult a qualified healthcare professional and verify regulatory compliance before considering any regenerative intervention. Now, let's stop the funny business. Let's start with biology, because biology doesn't care about sales funnels. Exosomes are extracellular vesicles, nanoscale lipid bubbles carrying proteins, lipids, and RNA. They're fragile, they're sensitive to temperature, pH shifts, mechanical shear, dehydration. That's why legitimate research labs store EVs at minus 80 degrees Celsius. Not because they're dramatic, because membrane integrity matters. Lyophilization, or freeze-drying, removes water. Without precise cryoprotectants like trehalose and validated protocols, it can cause vesicle collapse, aggregation, cargo loss, and reduce biologic activity. Yes, some lab studies show lyophilized EVs can retain structure under tightly controlled experimental conditions, but that is not the same as clinic-grade powder sold as plug-and-play injections. Here's the hard truth. There is almost no clinical evidence supporting lyophilized human-derived exosomes for patient treatment. What does exist? Mostly in vitro and animal studies. A few early-phase programs like platelet-derived EV products, PEP, under strict GMP conditions. No randomized trials proving that clinic-marketed lyophilized MSC or cord-derived exosomes retain full potency after reconstitution. What's missing? Large human randomized controlled trials, long-term safety data, post-lyophilization potency validation. If a clinic says this is proven, ask, where's the trial? The FDA has been clear repeatedly since 2019 and continuing through this year. There are no FDA-approved exosome products. The agency has issued warnings and enforcement actions after patients suffered severe infections, immune reactions, vision loss, tissue necrosis. Specific to lyophilized powders, published case reports describe granulomas, persistent nodules, skin necrosis, and scarring after injection. This isn't theoretical risk, it's documented harm from uncharacterized, unapproved biologics. Let's talk about what a lot of people are actually seeing on the ground, especially in places like Florida. Across the state, a growing number of regenerative clinics market placental-derived products. These are typically sourced from donated full-term placentas and may include material from amnion or chorion membranes, umbilical cord, tissue, or fluid, placental mesenchymal stromal cells, extracellular vesicles, or so-called exosomes, Growth factor-rich tissue extracts. Many of these products are processed, lyophilized into a powder, and then reconstituted with saline for in-office injection, IV use, or topical application. Clinics associated with large regenerative networks, as well as independent longevity or aesthetic centers, often advertise both liquid and lyophilized versions of these products, sometimes using proprietary centrifuges, filtration systems, or freeze-drying equipment as part of their processing pipeline. Now, here's where the confusion sets in. You will often hear clinics say that their treatments are offered under an IRB-approved protocol. That part can be true. An institutional review board, often a third-party IRB, may oversee patient registries, observational safety tracking, data collection on outcomes. But this is critical. IRB approval does not mean a product is FDA approved, FDA cleared, or legally marketed as a drug or biologic. IRBs oversee research ethics, not product approval. An IRB does not validate manufacturing quality, biologic potency, clinical efficacy, or long-term safety. So when you hear IRB approved, you should translate that as data is being collected, not this therapy is proven or authorized. Florida's regulatory environment has become more permissive in the recent years. Updates to state statutes around adult stem cell and birth tissue-derived biologics, combined with high demand and a wellness-driven population, have made Florida a hub for regenerative offerings that would not pass federal approval standards. This doesn't automatically mean every clinic is acting in bad faith, but it does mean oversight varies widely, products may differ batch to batch, and characterization often falls short of International EV Standards, or MISEV. The science checkpoint here. As of January 2026, there are no placental-derived lyophilized exosome products FDA approved. There are no randomized human trials confirming full biologic functionality after freeze-drying and reconstitution. Many products sold in clinics are not characterized pre- and post-lyophilization. Loss of membrane integrity, aggregation, and cargo degradation remain well-documented risks. Yes, there is emerging research. Optimized lab lyophilization with cryoprotectants like trehalose, platelet-derived EV products like PEP, and tightly controlled trials. But those are research programs, not mass-market clinic offerings. What's sold commercially is often not equivalent to what's tested in academic studies. So here's the bottom line, clean and honest. Lyophilized placental products may not be inherently useless, but they are far from validated. Patients are often assuming stability that hasn't been proven, potency that hasn't been measured, safety that hasn't been established. If someone is considering one of these treatments, they should demand full source documentation, missive-compliant EV characterization, potency assays post-reconstitution, actual human trial data and not testimonials, clear disclosure that the product is not FDA approved. Anything less is assumption-based medicine. If a clinic promotes lyophilized exosomes, ask all of these. Is this product FDA approved or under an IND? Show missive-compliant characterization before and after lyophilization. What potency assay confirms function post-reconstitution? Where is the human clinical trial data for this exact product? What is the adverse event monitoring plan? If they can't answer clearly, walk away. That's not advanced medicine. That's experimentation without consent. Bottom line in 2026, lyophilization is a legitimate research tool with future potential, but clinic-marketed lyophilized exosomes today do not have the human evidence to justify their claims. This isn't about fear, it's about biologic literacy. Regenerative medicine is real. Exosomes are real, but evidence still matters. Convenience doesn't replace validation. Powder doesn't equal proof. Protect your biology. Demand data. Stay signaling. Stay skeptical. Stay unbreakable. And tonight, I am finally out. Thanks for listening to the Human 5.0 Project. If you found value in today's episode, share it with someone who's ready to elevate their biology. Make sure to follow the show for weekly deep dives into peptides, protocols, devices, lifestyle strategies, and the science of high-performance longevity. Until next time, Stay curious, stay optimized, and stay 10x.